In our previous lectures, we have discussed the process of membrane transport and the features of ion channels. The transmembrane movement of ions provides a basis for electrical activities of the cells. Electrical activity is of great importance for living cells. Nerve conduction, contractions of cardiac, skeletal, and smooth muscles, and glandular secretion all depends on this vital cellular property. So this time, we'll talk about some of the most basic concepts related to the bioelectricity of cells. Here are the learning objectives today. By the end of this session, students should be able to define equilibrium potential and resting potential. Explain how the resting membrane potential is established and maintained. As you are now aware, the selective permeability of plasma membrane can lead to dramatic ion flows. When gated ion channels open, ions diffuse quickly across the membrane, following their electrochemical gradients, creating electrical currents and voltage changes, also called membrane potential across the membrane. Then, what is potential? The human body is electrically neutral. It has the same number of positive and negative charges. However, some regions may have more of one type of charge, making those regions positively or negatively charged. The difference in electrical charge between 2 points in a circuit is known as potential. It is measured in either volts or millivolts. Ion movements across the plasma membrane depends on the electrochemical gradient. If these gradients are equal but opposite in direction for a particular ion, then there will be no net current flow. This is called the electrochemical equilibrium. The voltage needed to oppose the flow of any given ion is thus called the equilibrium potential for that ion. For human neurons, the equilibrium potentials for potassium ions and sodium ions are minus 90 millivolts and minus 60 milliohms respectively. Resting membrane potential refers to the electrical potential difference between the inside and outside of the cells and the resting condition. It is also called membrane potential, transmembrane potential difference, or transmembrane potential gradient. The resting membrane potential is minus 90 in our skeletal muscles and is minus 50 to minus 100 millivolts, averagely as minus 70 millivolts in the neurons. You may ask, how did you know this? Physiology is an experimental science. Scientists got to know it through experiment. Squirt is delicious, but what you may not know is that squirt lends itself to research of neuroscience because its individual giant axon can be isolated very easily. Simply, one microelectron of the voltmeter is placed in the axon and the other in the extracellular fluid. A voltage across the membrane is recorded. It is approximately minus 70 millivolts. The minus sign indicates that the inside of the membrane is negatively charged relative to the outside. This potential difference in a resting neuron is called the resting membrane potential, and the membrane is said to be polarized. Two factors contribute to the resting membrane potential: differences in the ionic composition of the intracellular and extracellular fluids, and the differences in the plasma membrane permeability to these ions. Ions are not equally distributed in the body. The interior of the cell has a lower sodium ion concentration and a higher potassium ion concentration than the outside. Negatively charged proteins help to balance the opposite charges of intracellular cations, primarily potassium ions. In the extracellular fluid, the positive charges of sodium ions and other cations are balanced mainly by chloride ions. Although there are many other solids, such as glucose, urea, and other ions in both fluids, potassium ion plays the most important role in generating the membrane potential. Now let's consider the differential permeability of the membrane to various ions. At rest, the membrane is impermeable to the large anionic cytoplasmic proteins, very slightly permeable to sodium ions, but highly permeable to potassium. Cell membrane has abundant leakage channels. Those potassium ion flows down its large concentration gradient very easily. As the positive potassium ions leak out, the cell becomes more negative inside. The negative voltage counteracts the concentration gradient, trying to pull potassium ions back into the cell. Sodium ions dribbling into the cell make the cell just slightly more positive than it would be if only potassium ion flows out. At about -90 millivolts, the concentration gradient and electrical gradient for potassium ions are balanced. A negative membrane potential is established, and this is the resting membrane potential. It is the equilibrium potential of potassium ions. Because some potassium ions are always leaking out of the cell, and some sodium ions are always going in, you might think that the concentration gradients would eventually level out, and the concentrations of sodium and potassium ions on both sides of the cell will finally become equal. This will never happen because of an amazing built-in membrane protein, the sodium-potassium ATPase. This is an ion pump. This pump, which first expels 3 sodium ions from the cell and then draws 2 potassium ions back into the cell. By doing so, the concentration gradients for sodium and potassium are maintained, and the resting membrane potential is stabilized. Therefore, sodium-potassium pump plays a vital role for normal cellular functions. Abnormalities in the number or function of sodium-potassium pump are associated with several pathological conditions, such as heart failure. In summary, resting membrane potential refers to the electrical potential difference between inside and outside of the cell and the resting condition. It is generated by the differences in the ionic compositions of the intracellular and extracellular fluids, and the differences in the plasma membrane permeability to those ions. Resting potential is maintained and stabilized by sodium-potassium pump. Next time, we'll discuss action potential. Thank you, and see you next time.